All right, welcome everyone to my brief tutorial on using the shrink swell program I developed that was helpful for my cell cryopreservation research. There are definitely other programs out there that might be better for you, but just in case this is helpful, I wanted to share what I've been doing. When a plate of cells is exposed to CPA solution, we can be thinking about what happens at the cellular level here. When the cell culture medium is replaced with this CPA solution, that CPA solution has a lower concentration of water molecules outside the cell than inside the cell. So that causes a net efflux of water out, which acts to shrink the cell, i.e. lower cell volume. And on the other hand, the CPA concentration outside the cell is much higher than inside the cell, so that causes a net influx of CPA into the cell, acting to increase the CPA volume. And so the question here is, well, what happens to CPA concentration in the cell over time? What happens to the cell volume over time? And to answer this question, we'll look at what the prediction is using the program just to get a sense of the results. I'm showing here the input here and then the program's output here. So the program's input is can be understood as follows. So the black curve here is the extracellular CPA concentration over time. So from 0 to 5 minutes, it's at 0% of the maximum CPA concentration, and from 5 to 10 minutes, it's at 20% of the maximum CPA concentration. And then this 20% step has the corresponding prediction from 5 to 10 minutes. Just thinking through that, we have the cell volume in blue here. This 1 here represents 100% of the initial cell volume. So starting at 100% of the initial cell volume, the cell then shrinks down. This is because water transport exceeds CPA transport, so this water out happens faster than the CPA in, so the cell shrinks down. The CPA is getting in to some extent, but that's the dominant effect. And because of this shrink, the CPA that does get in gets increasingly concentrated because of that cell shrinkage. So that's the first part of this inclining curve here. And then the final shallower part of the CPA increase here is due to the CPA just continually permeating in here until we hit the 10-minute mark. And then there's another shrink swell event with the next step. So, the cell shrinks down again. That causes the CPA concentration to spike again, and so on. And you can go through a similar thought process to understand all of the steps there. All right, here is how to acquire the program and run it. So you can just go to my GitHub profile to download the shrink swell program that I coded. It's going to be this one, cryopreservation cell exposure to CPA. And the one you want to download is labeled final program. And you can download it by just left clicking on this download button. It's a Mathematica program, so you definitely do need to have Mathematica on your machine to run it. There is some other information on this website here that is not necessary but might be of interest. So in this developmental folder, you can see first the benchmarking program I did. So this is generating a same result that was published in the literature. And then here are a couple of corrections to that process to lead to a better result. So here, just fixing an error with how that published method was processing the molar volume of CPA. And then for units, consistency in the equations, doing everything using osmolarity. So that's those two to generate better plots. And then to actually extract some information out of those plots, I further developed the program to get stuff like the max and min cell volume. All of that is in the final program. This is just how I happened to get there. All right, and so if you open up this program, there is one section, which is this section, and this section contains an input and then various outputs. So this is the input, and then this input has various outputs. And just to give, this is just giving you an overview of how this is structured. And for this final output, which is a table, this message is going to appear. This is safe dynamic content, so I would click enable. And that gives you the data extracted from the plots. All right. You can read this information, though I'll walk through much of it. Again, the main thing we're trying to output here is what happens to the cell's intracellular CPA concentration and cell volume as a percentage of initial volume over time. I'm not going to go through all of these inputs, but I'll go through some of the ones that I think are less straightforward. So there are many inputs. There's this general inputs section, as well as there are more specific input sections here. And I'll go through all of them. I'm not going to go through like the processing of the inputs to get to the result, but I'll go through how to actually use the program. All right, so as you've likely observed here, anything that is a comment is in this gray text, and you can comment out sections of code using this here, this parenthetical and asterisk on either side. So stuff like this is not processed by Mathematica. This is not processed when the code is run. I guess I should not have highlighted that. So I guess if this happens, you can just click. So what I did is I clicked Alt period to abort. I think it was doing something weird. So yeah, the abort button, if anything weird happens, like there's a loop or something gets entered wrong, you might encounter that as Alt period. I think it's also in here somewhere. But anyway, you can click Alt plus period to abort whatever it's trying to do. All right, so those are how to understand the comments, and these variables are in the blue text here. So for this first variable, this profiles variable, you can enter many profiles. I have it set up for the entry of 3, each separated by commas, right? So there are 2 commas for the 3 profiles. One is this one-step profile, which is exemplary / instructive, and these others are profiles with more steps, which might be more practical, at least for doing a vitrification approach. And this one involves a singular change of using a longer final loading step time. So by running this code, we'll be able to compare all the other profiles to understand these profiles and to be able to customize them yourselves. Here is the information. So within each profile, there are 3 entries. The first is extracellular CPA concentration as a function of time. That's this whole thing that I've highlighted. The next is surrounding temperature as a function of time. That's that I highlighted. And then this number is the location of the final CPA loading step. Okay, so within the CPA concentration as a function of time information, here are the units. The first is percent. So this is 100% CPA, so that's the max CPA concentration, versus 0% CPA. That's this first entry here in each of these steps. So this is step 1 of the profile. This is concentration step 2 of the profile. And then the second entry is the time, the duration. So here it's going from less than 5 minutes. The units of this is seconds, so 5 times 60 is 300 seconds or 5 minutes. So this is a single loading step to start things off for 5 minutes. And then here for the next 5 minutes, it's going back down to 0% CPA. I said for the next 5 minutes because where this 0% is actually taking effect is you can think of it like this. It's between 5 and 10 minutes. But the way that the information actually gets processed, it should be written like this, where the sort of prior step is left off and kind of implied there. So that's how to enter the information there. Similar for entering the temperature information. This is in Kelvin, so we're going from a 4 degrees Celsius step to a 22 degrees Celsius step throughout that process. And then, yeah, a similar sort of thing, just, more entries for all the different concentration steps here. And then it looks like I probably used the same, or very similar at least, temperature profile, where I only have it running at room temperature for the very last step. And you can see that the final loading step here is 5 versus 1. So the first step here, which is at 100%, is designated as the final loading step. I would expect everyone using this program to do that, given the application of cell cryopreservation. But there is a case, I suppose, you could make for not having it be, whatever the max concentration is. So here it's 1, 2, 3, 4, 5. So I put it as 5. So for example, the 2018 Benson article has shown is that you might actually want something like this for the application of tissue cryopreservation, where you want like this bigger concentration step to precede the final CPA loading step. So in which case, you would actually have it be this. But I think probably for cell cryopreservation, we wouldn't want that. So I'm just going to change it back to how it was before. All right, and so that's how to understand it. And then, I think once you understand it, you'll be able to customize these how you want, comment these out, delete these, whatever, to test various profiles, see it to one you're satisfied with. A lot of these, these inputs are going to be system dependent. So just think about what you want for your system, test different parameter values, all that sort of thing. If you want to export these data, enter 1 for the volume versus time data, and then 1 here if you want intracellular CPA concentration versus time data. And then, going into the next section here, this gets more complicated depending on what the user wants. So if the user wants osmolality inputs, that's a second section. What the user should do is comment out the osmolarity section and remove the commented marks around the osmolality inputs, and then enter their stuff in terms of osmolality. So this would be the osmolality of the CPA in the CPA concentration, or in the CPA solution, and here would be the osmolality of the carrier component of the CPA solution, which is of course analogous to the osmolarity inputs here. This information is 1. This should be set as 1. This should be kept at 0. This is just going to let the code process as it should to convert all this from osmolality to osmolarity if you do choose to input things in terms of osmolality. All right, moving on to the model geometry. So I used a hemisphere model, which I'll keep, but if you don't want that, just comment out the hemisphere model, and you can instead have it, for example, in this case, use a spherical model instead, or you can choose this elliptical cylinder model. So those are the different types of models available. And then finally, this is solved using the 2-parameter formalism. 1 parameter for the water permeability, 1 parameter for the CPA permeability, though it might be best to think of these as composed of more than 2 pieces of information, because there is this temperature dependence here that I think should be accounted for whenever you do this. So for example, if you know that your system has a hydraulic permeability of 5 times 10 to the negative 14th at 22 degrees Celsius, so this is the permeability at this temperature, you should absolutely, customize that accordingly. I'll just leave it with what I have. And so, yeah, I think we're done explaining it. I'll just click run here. And to cause this program to run, you can really left click anywhere inside of this input code block and then click Shift Enter on your keyboard to run. When you do that, this is going to be blacked out, and various pieces of output will populate and will replace the prior outputs. So, if you wanted that prior information, say you were changing something, definitely save this information first. But at any rate, what the output is demonstrating here is that we're going to be testing various profiles using these parameter values. So these came from the temperature-dependent parameter values that we had entered before. This is just giving you a sense of how fast or slow the permeability values you are. Only really meaningful if you've done this a lot. But at any rate, theories are the CPA exposure profiles. Simple one here, just a one loading step followed by one unloading step, and then we're testing more complex ones later. Yeah, you can see that this code could be improved so that when we overlay these curves, it doesn't chop it off. But at any rate, I think it's good enough to be useful. And so here's basically the information I like to look at, which is this final table here for comparing them. So here looking at the minimum cell volume. And so with this first profile where we don't have any loading steps prior to the final loading step at 100%, it has the lowest minimum cell volume. Maybe that's too much cell shrinkage for your system, don't know, but it's certainly more risky in that regard, and that's you can visualize that here with that blue curve dipping the lowest. And then on the other hand, these other profiles seem to have more risky cell swelling. So there's cell swelling to over 2 times the initial cell volume, whereas the more simple profile with the one unloading step has less of that. I think that makes sense because you're starting before the unloading step for the simple profile. You're starting at a lower volume to begin with, whereas here we're starting at a higher volume to begin with before the unloading. And also these unloading steps, I think, are all at 4 degrees Celsius for these profiles until the very last one. So only at the very end of each profile, like this loading step for the blue profile is at 22 degrees Celsius. Permeability is much faster at 22 degrees Celsius, so that helps the cell get back to a healthier cell volume quicker. So yeah, I think that makes enough sense. Definitely something to understand, and you can understand this rather well just by experimenting with different profiles here. And then finally, you get the potential benefit of extending the CPA loading time from like 5 minutes to 25 minutes here, as you get a bit more CPA equilibration, 97% versus 93%. Not a huge difference, definitely diminishing returns on that. And yeah, that's the trend here. And that's how to understand the output and to hopefully be able to edit or customize the inputs yourself and run the program.